Okay, so good afternoon. I think you need to start again because this is one of the most important sessions of this conference. I heard that I was provoked this morning, but again, this is all about immunology, French, Brazilian immunology. So we have a very nice presentation from four great scientists, what we call young scientists here. But I have to say that we need to keep on time each presentation because after that we're gonna go to the ending and then people need to go to the airport and so on. So it will be very important if we keep time and someone will help us. So I'm very glad again to be here to present four great scientists that I met throughout my from the last couple of years. Each one have a very interesting story to present today, more or less supported by FAPESP, and this is very important. So first I'd like to call Tathiane Maistro. She's a scientist, a research scientist at the Faculty of Pharmacy and also Faculty of Medicine at Ribeirão Preto campus. She won a L'Oréal Prize a couple of years ago and is a young scientist supported by FAPESP. She's working on epigenomics and brain tumor, and for us it's just a big pleasure to have you here, Tathiane, please.
Eu pode passar. Tá bom. Good afternoon, everyone. So I'm more well known as Tati Malta. So my name is Tati Malta, and I'm very happy and honored to be here today. I'm a FAPESP Young Investigator grantee. This is a very special opportunity for me to establish my own group. I'm located at campus of Ribeirão Preto, as Niels said. And my lab has mostly working on computational biology to understand molecular mechanisms of cancer, mostly brain tumors. So, in other words, basically what we do is trying to tackle the precision oncology. So according, and I'm going to show today what we are working in this field and how my lab is contributing to precision oncology using multi-omics data, as I said. So according to NCI, precision oncology is when you use information from the person or from the tumor to help improve either diagnosis, treatments, and prognosis. I've been working with adult gliomas for the past years. So what we have right now, according to the WHO last update on adult glioma classifications, so every tumor should be classified based on either both histopathological and genomic. So an adult glioma should be divided further between according to IDH status. So we have IDH1 and 2 mutant tumors and IDH wild type tumors. The IDH mutant tumors can be further subdivided between chromosome alterations, most 1p19q intact chromosomes, or the presence of the co-deletion of 1p and 19q. So that's what we have so far, and this is have a very good correlation with clinical prognosis. So the IDH wild gliomas is a very aggressive tumor type, right? So the IDH wild type is the most aggressive one, and those are known as glioblastoma or astrocytoma grade 4. And the ones with IDH mutation, they are the lower grade gliomas and have a better prognosis. The ones with the co-deletion, the blue group here, they have the best, the most favorable prognosis, if you can say so. So my story in the adult glioma field started 10 or 11 years ago. I was doing my postdoc in Ribeirão Preto and later on in the US. We are working as part of the Cancer Genome Atlas, or TCGA, analyzing epigenomic data of adult gliomas. So at that time, I published this paper where we defined 7 subtypes of glioma based on DNA methylation. And the most important finding in this paper was that IDH mutant that I just told you that have the better prognosis. We identified actually 3 subtypes, and based on DNA methylation and this special and previous unknown subtype that we named G-Simp low because it's lowly methylated compared to the G-Simp high, which are the other IDH mutant tumors with a better prognosis. So we propose that the IDH mutant 1p19q intact adult gliomas should be further stratified based on epigenomics in G-Simp high and G-Simp low. G-Simp low with a more aggressive behavior and should be managed probably differently, right? So after this study with the CGA, I joined the glioma longitudinal analysis where we collected glioma longitudinal samples. It is an international consortium with some samples, including from Brazil. And we had a total of 77 IDH mutant gliomas, and I'm from now on talking about IDH mutant gliomas. And we found that 20% of the IDH mutant patients actually switched subtypes after the upon recurrence, right? So they are the tumors change their phenotype to a more aggressive. So for some reason, some of the tumors recurs is the same epigenomic phenotype and clinical phenotype, but some of them progresses to a more aggressive behavior. So we start trying to understand why this was happening, and we found that actually basically half of the IDH mutant gliomas, they received treatment beyond surgery, and some of the patients, half of the patients did get any treatment besides surgery. When you looked into the recurrent tumor, so after the surgery and the tumor recurs, we found some regions in the genome where the tumor were lost methylation, so they were lowly methylation, the treated patients compared to the untreated group. So those tumors at primary stage, they have high levels of methylation, and again, the ones that have been treated, they lose methylation for some reason, and in these regions, the same genomic regions were already lowly methylated in IgA wild type, both at primary and recurrence. Again, those are the most aggressive phenotype. And when we looked into a more extended cohort of IDH mutant gliomas only, dividing by treatment status, we found out that actually when you divide IDH mutant treated and not treated, the treated patients actually have a small bad behavior. But however, when you looked into the surgical interval, the treated patients actually the tumor took long to recur. So the treatment somehow was delaying the tumor recurrence, which is what we expected, right? But however, when I looked into the overall survival from the second surgery to the last follow-up, the tumors the patients that didn't receive any treatment, they looked better. So in summary, the untreated IDH mutant patients had a shorter surgical interval, those tumors recurred faster, but overall, in the long term, they lived longer. This was kind of interesting. According to the neuro-oncologists from the group, it was validated in an independent cohort, and these results was published. Those are all done here in Brazil as part of my FAPESP grant, and was just published a few months ago, and we saw that after the standard treatment of those tumors, we see a loss of DNA methylation, epigenetic activation of important genes, changes in the microenvironment, and potential acceleration of tumor progression. So this was all retrospective. So of course, now we are designing some experiments and plans to address those. And FAPESP just gave me an extended grant, extra money to perform some experiments, both in vivo and in vitro. So besides this project, I have other sub-projects in my group. I just want to briefly highlight this project on tumor systemness. So as part of the TCGA, we published in 2018 this paper where we defined algorithms to measure stemness in tumor samples based on transcriptomic and proteomics. And now we are working on expansion of this project where we are deriving algorithms based on proteomic. So we derived the ProTSI. It's currently under review at Cancer Cell, and I'm a co-PI of this project. And with this algorithm, algorithms, we're able to identify some proteins predicted by stamina status, and they also are predictor of survival, and some of those proteins can be targeted by non-drugs. So we also, part of this project, we validate by immunohistochemistry some of those markers with correlation of survival. So this is a whole another project that I wanted to show here. It is unpublished yet, and this I have other algorithms to predict staminas and other phenotypes in the lab. So basically, we are trying to predict staminas in tumors across a pan-cancer fashion. So those are the results that we have been working in the lab, right, to improve diagnosis, treatment, and prognosis. I hope I can convince you. And again, my lab is very collaborative. We have collaboration with several international groups. Unfortunately, not with France yet. I hope we can build something in the future. And that's it. Thank you so much. Thank you again, FAPESP, for this opportunity. And I'm open to questions.
Tati, very nice last picture with a lot of beer. So just to make a proof of concept here right now, you see, our next speaker was once a day, long time ago, a physiologist. And then he turned into immunologist because he saw that the future will be brighter. So I'm very glad to introduce Professor Marco Vinolo. He's associated professor at UNICAMP, the State University of Campinas. He's a well-known scientist here working in microbiota and immunology. So thank you very much.
So good afternoon. It's really a pleasure to be here. Thanks Niels and the organizing committee for the opportunity. I'm a physiologist but I'm working with immunology and microbiology, and I'm happy to show you some of our results. So we work in this interaction between host and microbiota, and for this presentation, I choose some of our recent papers to give you an overview of what we are doing, what are the tools in our interest. So I hope I can give you some ideas of how also to collaborate with us. And just brief introduction. So all of you probably heard of microbiota. I don't need to go in further details, but I just would like to emphasize that despite the advance in the last few years, we still don't know a lot about these interactions between these microbes that colonize our body and our own cells. So my lab is interested on understanding how these interactions happen and how we can affect these interactions and promote health or avoid development of disease. We know that there are several factors that interfere on this interaction, and here I listed two big groups of factors that act in the system, so environmental and host genetics. Of course, the genetics of the microorganisms is also other important point, but I choose these two to show some examples of studies that we have been developing in the lab in the last few years. So we know that when we talk about microbiota, one of the first things that come to our mind is the gut microbiota. And one of the factors that regulate the gut microbiota is diet. So we have been working with dietary fibers. So these are molecules that are used by several components of the gut microbiota, mainly bacteria. And we know that the bacteria use these molecules to produce several types of molecules which act on the microbes itself and also on the host cells. So we used one type of soluble fiber in this study that I will show in the next slide, which is inulin. So it's this molecule. It's a polymer of fructose. And what we found, and it was published last year, is that this molecule causes very interesting and dynamic effects in the system, the gastrointestinal tract. So we have seen that this diet interferes with the microbiota composition and also functionality. And we demonstrated that this effect affects the macroscopically the tissue. So we have a longer large intestine, but also histologically, as you can see here, we have a different disposition of the crypts. I won't be able. So there is a difference in the depth of the crypt, and also we saw increased proliferation of intestinal stem cells. The interesting thing is that we saw that not only the microbiota is necessary for this phenotype, but also immune components as interleukin 22 and also gamma delta T cells, which we are now investigating in more details. We using the same type of diet in mice. We have seen previously that this diet can also protect mice from enteric infection. So we worked with a model of infection caused by Clostridioides difficile, and we demonstrated that this diet caused relevant change in the microbiota, which in this model has a protective effect. We also found using then other type of fiber, interestingly, that there is also other mechanisms by which it's not appear, but we found with other type of diet also a protective effect in this model, but through a totally different mechanism. So in the previous slide, I showed impact directly on the intestinal epithelial cells through butyrate production, and here we demonstrated that through production of acetate, which is other short-chain fatty acid, we have a coordinated response involving type 3 innate lymphoid cells and neutrophils. In both cases, we have protection. In addition to that, we have seen many other factors that interfere in this interaction. And here is other study that we published a few years ago, in which we look at for genetic modifications in intestinal epithelial cells. And in this study, we demonstrated that antibiotic treatment caused a drastic change in the production of short-chain fatty acids and also on several other molecules. And this interfere with the histone modifications on intestinal epithelial cells. So in this study specifically, we demonstrated that crotonylation, which is one type of acylation, is sensitive to microbiota. And we are now exploring this in more details. This is other study that we plan to submit soon, but it's in the same line. So I'm showing here other study in which we demonstrate that the microbiota depletion with antibiotics not only affects what the cells are expressing, but the population of intestinal epithelial cells or the subpopulation of these cells in the tissue. So in this study, we are describing a population of hybrid absorptive secretory cells, which seems to be increased after depletion of microbiota and is also seen in other types of dysbiosis. We saw the mechanism by which this effect is caused, and now we are closing the gaps to submit, and hopefully we will publish this in the next few years. Other thing that interfere on this interaction is infection. So here just an example of a study that we conducted together with Ana Paula Duarte de Souza from Rio Grande do Sul, in which we analyzed the microbiota of kids infected with RSV, which is a respiratory virus. So we've seen that this infection not only caused changes in the composition of the microbiota, but also cause changes in the function of this microbiota. And we, using experimental model, we demonstrated that reversing part of these changes protected mice against this infection. More recently, we collaborated with François Troutin in a very nice study that we published this year, in which we analyzed the microbiota with in a flu infection model. And one thing that caught our attention was the drastic change in the concentration of one metabolite called IPA. And this metabolite is linked with tryptophan metabolism. And what was interesting was that not only we found this difference in mice infected, but also when we treated mice with this type of molecule, we have a better response to the infection with lower virus load and also less inflammation in the lung. Next, this is a study that we are also conducting with François Trotton, but in this case, we are seeing the infection caused by SARS-CoV-2, and we've used two different models. So we used aged and young hamsters. So we know that the microbiota is responsive to the infection by this virus, and we wanted to understand how this response is affected by the aging of the host. And as you can see here, we demonstrated that aged hamsters, they have worse response to infection, and this is directly associated with more intense and drastic changes in the microbiota composition and also in function. But I will not have time to show all the details. Other factor that we explored a few years ago is the oxygen level. So we demonstrated through different studies that oxygen in the gut is very important. It regulates not only the microbiota composition, but also the activation and the function of different cells present in the intestine, including intestinal epithelial cells, but also including other cells as shown here for type 3 innate lymphoid cells. So the other hand, we have the genetics factor. So we have been studying mainly with murine model, many factors that interfere on this, that potentially interfere on this interaction. And here I would just call your attention for the two genes that we studied together with other groups. So SMARCAD1, which is a chromatin remodeling factor. So we deleted this gene in intestinal epithelial cells, and this caused a change in the microbiota, in the gut microbiota. But in this case, this change was protective against colitis induction by DSS. In another paper that we published recently, we demonstrated that deletion of other factor, in this case IL-22 binding protein, in intestinal epithelial cells is also associated with drastic changes in the microbiota composition, and these changes are protective in colitis model and also in infection model, demonstrating that not only environmental factors, but also host genetics is essential for this balance. So I would like just to finish by saying that we are exploring many other aspects of this interaction, and we using different tools for that, and we are more than happy to establish new collaborations. So please let me know if you want to discuss further any of our results and other possibilities to interact. I would like to finish by saying thank you to all the people that worked with us, including Niels and many other researchers from here, Brazil, and also from other countries, and of course, the funding agencies that support our work, especially FAPESP, and the students and researchers that worked with us and made this possible. Thank you.
As you can see in the last slide, there is no beer in Campinas. So if the students want to stay, it's better to stay in São Paulo than go to Ribeirão Preto. So thank you, Marco, wonderful presentation. So the next speaker is someone that I met a couple of years ago, and I got really impressed by all the ideas and also the way she is as a person. So I'd like to introduce Dr. Carolina Alves Costa Silva. She's associated research at the Department of Immunology of the Institut Gustave Roussy. So she'll talk a little bit about gut microbiota and oncology.
Okay, so I'd like to start by thanking you for the invitation. It's really a pleasure to be here. And I'm going to present actually my thesis during the last 4 years that I was able to defend last year. I cried while I was preparing my slides, and it's about gut microbiota-related biomarkers in immuno-oncology. And just to start, microbiota controls the brain, that controls immunity, so that's why I chose this field. So from a better understanding of the tumor microenvironment, we could develop a lot of biomarkers in immuno-oncology, but today, unfortunately, a few of them pass the validation phase, and only 3 have FDA approval to date. The impact on the microbiota on cancer immunosurveillance in our group were demonstrated using 4 proofs of concept. So first, the impact of antibiotics. We looked in retrospective and prospective cohorts that patients that took broad spectrum antibiotics, they had worse survival. Then we could see by doing metagenomics shotgun that patients that respond, they have a different gut microbiota composition from patients that don't respond to immunotherapy. We could validate that in avatar mice, that when we transfer the feces of patients that responded, these mice were sensitive to immunotherapy. And now that is the most exciting part. We have these pilot studies that are out and showing that microbiota-centered interventions are promising in non-selected individuals. And why non-selected individuals? Because in this complexity, I came to add even more complexity, proposing these gut-related biomarkers to select these patients in clinics. So we have demonstrated that cancer induces a stress ileopathy, and then it's followed by a gut dysbiosis that is long, and this gut dysbiosis participates in tumor progression. First, we had demonstrated in mice, then we could see that in humans, in patients with cancer, they have a different gut microbiota composition compared to healthy volunteers, with a dominance of this species associated to the enterocluster genus. And we know that already showed patients with cancer have a different microbiota composition from healthy volunteers, but when we look responder and non-responders, we see that patients that respond to immunotherapy, they have a gut microbiota composition that is more similar to the healthy status. So to see, not all the patients will need microbiota-centered interventions, so how do we define dysbiosis to be able to select these patients in clinics? In dynamics analysis, we have demonstrated that these patients that really respond to immunotherapy, and here we have selected the patients that had a complete or partial response lasting more than a year, and an overall survival of more than 2 years, showing that the responder patients, they lose the enterocluster, the tolerogenic enterocluster, while the patients that didn't respond, they lose the immunogenic Faecalibacterium prausnitzii. And our favorite bacteria, concordo e discordo de Renato, showing that Akkermansia mucinifila, a specific strain using metaflam-4, we show that normal levels, and by abnormal I say patients that have a higher relative abundance or that are negative, so patients with these lower levels of Akkermansia mucinifila, they have a better survival. And when we look at the gut microbiota composition, we show that Akkermansia can be a proxy of gut dysbiosis because patients that have these abnormal levels, they have a gut microbiota composition that is more tolerogenic, and these patients, they have these higher levels of exfoliated genes, so maybe mirroring the stress ileopathy that I have demonstrated before. And due to the limitations, if you look all the studies around the world, we can see different species that are associated with responder and non-responders. So we try another strategy, and we built this network based on ecological distribution, and we could identify the bacteria that are associated with response and non-response. Since yesterday, I had in press, like in the reference, but the paper is out today, so I hope you enjoy reading. And from this whole population-based network, almost 500 patients with lung cancer, we could compute this unidimensional score that could be translated into a PCR, so done in 24 hours test, to be able to select the patients that have a bad gut microbiota composition and would need microbiota center interventions. These are the patients that I call SIG1+, that are in black. And it was really interesting to see that applying Topo score to a big database of healthy volunteers, we could see that 25% of these healthy volunteers, they harbor gut dysbiosis, showing that this strategy and all the biomarkers that we are developing can be used in the interception strategy to identify people that are maybe more potential to develop some inflammatory disease. And knowing that gut dysbiosis is not cancer specific, so this beautiful paper by Gacheza showing that different inflammatory diseases, they share the signature that is tolerogenic and it's gut dysbiosis. And one of the more beautiful paper that I had the chance to participate in the lab. So we showed the mechanism associated with the antibiotics deleterious impact, showing that it disrupts the intestinal regulation of this lymphocyte migration through the gut immunocheckpoint MADCAM1. So this MADCAM1 is expressed in the high endothelial venous, and we show that after antibiotics, we have the enterocluster that appear. These enterocluster, they change the metabolism of the gut, so more bile acids are going there. And then we have the reduction of the MADCAM, and these Treg cells, TR17, they are able to migrate into the tumor and lead to cancer resistance and progression. And really nice, we are talking about biomarkers. We could see that correlating with these ileal transcripts, the soluble MADCAM could be a prognostic biomarker in cancer immunotherapy, and also a proxy of gut dysbiosis, since patients that had lower levels of soluble MADCAM, they have worse survival, and also a gut microbiota composition. You can see in orange, you have Veillonella parvula, enterocluster, so tolerogenic bacteria associated with low MADCAM. And it was not only in these 3 independent cohorts, now we have newer ones that are not published. So patients with localized breast cancer, renal cell carcinoma treated with immunotyrosine inhibitors in first line, and also colorectal cancer treated with chemotherapy with or without atezolizumab. Patients with high MADCAM, they also have a greater survival. So showing that cancer progression, they lead to this stress ileopathy, then to gut dysbiosis, so you have the enterocluster that goes out. It leads to metabolic changes, down regulation of MADCAM, and then migration of these immunosuppressive cells. And we could show that, demonstrate these impacts of the changes in metabolism in patients with localized melanoma. We could see a shift months before the recurrence in patients that had a relapse of the melanoma afterwards. So these patients, they had this accumulation of biliary acid composition, acetylated polyamines, and also a signature that you already know that is associated with immunosuppression. Enterocluster, Veillonella, so all the bad guys. And when we integrated both metagenomics and metabolomic signature, we could better stratify the patients regarding the prognosis. So in this case, medium and long chain acylcarnitines that were associated with relapse. So patients that have lower levels but higher levels of Faecalibacterium prausnitzii associated with a better prognosis, these patients, they went better. Going to another biomarker, so there is already evidence showing that microbial immunity can elicit anti-cancer immune response. And indeed, we have showed that circulating humoral response, so IgG titers against E. coli, and also that these E. coli were capable of releasing CXL13 in the PBMC of the patients. These were correlated with benefits to PD-1 in patients with localized bladder cancer. So here I have these 4 omics associated with biomarkers. So taxonomic, gut barrier fitness markers, metabolomics, and also immunomics. And now what happens when a patient arrives at the Gustave Roussy? They are proposed to be included in the clinical biome, our prospective study. And we see them in clinics to try to evaluate all the factors that are associated with the risk of gut dysbiosis. And also they are seen by a pharmacist and a nutritionist to evaluate all the factors that are associated. And we are able to prospectively validate all the markers that we propose. And sure, we have these staff meetings because now you have clinical trials that are open. For example, for patients today that have Akkermansia mucinifila that is negative, he can be included in the EV2101 clinical trial to do Akkermansia as a probiotic in the first-line setting of patients with lung and renal cancer. And also, we continue to collect our omics to try to understand the pharmacokinetic and pharmacodynamics, and also develop new therapeutic strategies. So I want to finish by thanking all the patients and caregivers that participated to the study, and all my rich scientific commensalism that helped me to finish my thesis, be able to change a little bit of the clinical practice, at least at the Gustave Roussy, we are doing something, hope we can spread abroad. And I'm happy to say that my thesis has been selected by University Paris-Saclay for the Prix de la Chancellerie. So if you are here, you vote for me, please. Thank you.
She will provide the link later on, so you can just scan the QR code and support her. So thank you very much, Ana Carolina. So now, just to finish this this session, it's my pleasure also to call the last speaker, Dr. Tiago Medina. He was a former PhD student on immunology at the Faculty of Medicine in Ribeirão Preto. He's now a group leader in Camargo, is a hospital related to cancer here in São Paulo. So thanks again, Tiago. Great pleasure.
Thank you, Niels. Thank you, Leandro Collier, for the kind invitation. I'm quite happy to be here sharing some data from my group of you. So yeah, I'm the leader of the Translational Immunology group at the Camargo Cancer Center. Let me see if I know how to use that mouse. Yeah, I can see here, but I cannot see there. Oh, there you go. I guess it's here. Is it working? Not really. But what about the laser, the pointer? Yeah, we cannot see there. Oh, there you go. All right, so yeah. So I think the whole idea is to share with you what we've been doing at the Camargo Cancer Center. And we have essentially two research lines there that we've been developing that are quite established already in the laboratory. The first one is about this beautiful structure here that is depicted in the screen, which is called tertiary lymphoid structures. That's essentially, can just move on. So that's essentially a beautiful aggregate of lymphoid cells. So basically, it's quite a consensus nowadays that tumor antigens and inflammatory mediators can essentially trigger lymphocytes, and this, after this activation, these lymphocytes can being aggregated in these beautiful structures that resemble a lymph node, essentially. So that's a very organized aggregate that are essentially composed of a beautiful B cell zone that are usually posed in the core of this structure, and that's usually surrounded by a zone of T cells. And that's quite important because that essentially promotes cell-to-cell interaction, and essentially the T cells and B cells get benefit from that, and that essentially induce immune responses against tumor cells. So we've been quite interested to characterize this structure across tumor patients. So we have quite a lot of single-cell data that we have been mining in the laboratory. So just to make a long story short, we've been showing that T cells, especially the conventional one, ones, not the regulatory T cells, they are able to make CXL13, which is a chemokine that essentially attracts whatever cell that expresses XR5. And when we look at the cells that are expressing XR5 or single cell data across all of the cells that we identified, we're able to identify that only B cells are able to express XR5. So that somehow suggests a possible interaction between BNT cells, within the tumor microenvironment of pancreas, pancreatic ductal adenocarcinoma patients. We essentially calculated that to make sure that the B cells and T cells could be indeed interacting. And whenever you do this calculation, just calculating the likelihood of interaction between a given cell that expresses XR5 and a given cell that expresses XL13, you can see that the only possible interaction is between B and T cells, either conventional CD4 T cells or CD8 T cells. When we focus only on the interactions between B and T cells, we can see that the XL13 and XR5 axis is the most important axis in this interaction. And just to make sure that we're talking about TLS, we essentially validated that, and we're able to at least to suggest here that this interaction could be happening within the tertiary lymphoid structures of these patients. That single cell data, it's quite important to validate that using, so we are always using omics data to generate quite a lot of data, and they're always validating that using immuno-based assays. So yeah, I'm an immunologist by training, so I do prefer immunology than oncology, Niels. So we could generate data like this one, where we essentially show beautiful TLSs in these patients, where we can see a beautiful aggregate of B cells in the core of this structure, as I mentioned before, and the T cells surrounding the zone of B cells, either CD8 or CD4 T cells. And essentially, we can see that the B cells exclusively express XR5, whereas the T cells express XL13, which again denotes the idea that this axis is quite important for the formation of this structure in these patients. There's another example here showing another mature tertiary lymphoid structure, and we could see that whenever a T cell zone leaves, a T cell leaves the T cell zone and invades the B cell zone, that somehow induces a phenotype that's associated with tumor reactivity. So we decided to validate that, and indeed we show that the T cells that are organized within this structure can induce tumor reactivity instead of, whereas the cells that are not organized in this structure, they cannot generate this phenotype associated with tumor reactivity. I won't go through all of this data, it's quite a lot, I don't have much time. But essentially, well, again, we are always validating these findings using immuno-based assays. We used quite a lot of flow cytometry just to essentially validate everything that we showed in single-cell RNA-seq data. Something that is quite important for us is the fact that when we do pan-cancer analysis, we could see that XL13 positive T cells across quite a lot of tumor types express exactly the same phenotype, which makes them quite conserved across all of the tumor types that we analyzed in this pan-cancer analysis. And something that is quite important is the fact that whenever the T cell is organized in the TLS, which is the XL13 positive clones over here, just by looking at the single cell TCR repertoire of the cells, we can see that these cells are much more polyclonal, indicating that these cells are expanding quite a lot. And essentially here, we can see that, oh, the mouse is not working. There you go. That the cells that are organized in this structure, they acquire this phenotype associated with higher expression of XL13, and this deem that it's much more associated nowadays with tumor reactivity. So bringing this idea that the TLS is quite important for the generation of anti-tumor responses by T cells, for the expansion of the T cells, and they acquire this phenotype associated with the immune response against tumor cells. We were quite interested to understand how the maturation of the TLS could impact the functionality of this structure. So we classified over here TLS across various degrees of maturation of this structure. So we essentially identified TLSs that are not mature, they're fully immature actually, intermediate stages of maturation for TLS, and TLS that are fully matured, and we just control that based on the expression of CD23, which stands for follicular dendritic cells. So we decided to microdissect all of these structures, and we performed gene expression in all of these different stages of TLS differentiation, maturation, and we essentially identified 6 gene modules associated with all of these stages of maturation. 2 that are expressed here when the TLS is still immature, and that's sustained across time or across the maturation of the TLS. 2 that just showed up when the TLS is intermediate, and that's sustained through the full maturation stage of the TLS, and 2 modules that are exclusively expressed by the mature TLS. And whenever we look at these genes, we can find that essentially the immune response associated with tumor reactivity only takes place when the TLS is fully mature. So we need this structure to be fully mature to generate anti-tumor responses against tumor cells. So we decided to project, again, our tumor reactivity signature onto all of these maturation stages of the TLS. And again, we could find that the anti-tumor responses only take place when the TLS is fully mature, just confirming the previous data. And we believe that this occurs because the T cells and B cell, the T and B cell interaction is only possible when the TLS is fully mature. And those are the interactions that are possible between these populations when the TLS is fully mature. If the TLS is not mature enough, we don't have any interaction between B and T cells, essentially. We just characterized the biological relevance of this structure in pancreatic ductal adenocarcinoma patients, and we decided to analyze the clinical relevance of that, obviously. So we took advantage of the TCGA cohort, and we essentially identified quite a lot of, actually, we're able to classify the patients according to the inflammatory infiltrate of these patients into 3 groups. The patients that have the formation of the TLS, when we look at the T and the B cells, the B and the T cells have a clonal expansion that are quite accelerated over there. And then we're able to identify TLS that are mature enough and TLS that are not immature. So we decided to generate 2 minutes, okay, a molecular signature that resembles this full stage of maturation of the TLS, and that was associated with better prognosis in these patients. And it was also associated with a better immune response to immunotherapy when we used this public cohort, the PRINCE cohort. So we are working on that, not only in pancreatic ductal adenocarcinoma patients, but across several tumor types. We actually have data that are more beautiful than this one, but they're not published yet, so I didn't bring it here. But the whole idea that when the cells are organized within this structure, everything happens. We have a better immune response against tumors, we have a better prognosis, and that's associated with improved response to immunotherapy. In these type of patients and across quite a lot of patients that we've been studying in the laboratory, we were able to write a comment at Nature Reviews Cancer about this structure last year. We're quite happy because that was the 3rd most downloaded and most cited comment in Nature Reviews Cancer last year. The editor told us that, so we're quite happy in the laboratory. And just to be quite quick, just to bring, I won't cover any introduction about that because Marion yesterday did this job, but we're also interested to understand or identify ways to improve the CAR T cell responses in our laboratory. We're also using epigenetic therapies. I'll just speed up my presentation, so never mind about that. But essentially, we're identifying some epigenetic therapies that are generating CAR T cells that are less differentiated CAR T cells that are associated with this beautiful phenotype associated with stem cell-like memory T cells. And we're quite interested about that because these are generating quite beautiful response against tumor cells. Whenever you look at phagocytosis is when the T cells try to fight a cancer cell and they exchange fragments of membrane. So that's quite harmful for the T cells because sometimes by exchanging fragments of membrane, the target of the tumor cell comes to the CAR T cells, and another CAR T cell can kill these CAR T cells that underwent phagocytosis. And we can see that our epigenetic therapy can essentially abrogate all of this phagocytosis processes in the cells. We can see that the CAR T cells are much more efficient in killing cancer cells compared to the conventional CAR T cells, and they proliferate quite a lot, even if you look at the total CAR T cell population or only within the stem cell-like memory phenotype. We are doing quite a lot of bulk RNA-seq on this population, and essentially the cells improves quite a lot when we do our epigenetic therapy. The cells proliferate quite a lot. As you can see here, the program is here. They are much less pro-apoptotic. They are very anti-apoptotic. They induce pro-tumoral, pro-inflammatory response rather than more pro-tumoral responses. They are less terminally exhausted, and they do not or polarized towards regulatory T cell phenotypes, and they are very migratory. So here we establish all of the good qualities about the CAR T cell. So they are much more antitumoral. They are very migratory, and they are less differentiated than the others. And they're also doing exactly the same thing in patient-derived CAR T cells and showing how we can potentiate the immune response of these cells as opposed to a more terminally exhausted phenotype. And the cells can expand quite a lot, and they can kill much better tumor cells in vitro, and they expand quite a lot, as we can see even in the microscope. So I'm going to stop here. Thank you, all my collaborators. Thank you, FAPESP, for the whole support. Thank you, CNPq, Ministério da Saúde. Both agencies are also helping us quite a lot. So thank you so much for your attention.
Thank you, Tiago. So I invite all the speakers to join us here. We're going to have a very quick session of Q&A. But just, I will ask something very strange, but very kindly. If someone has an Apple AirTag, if they could switch off, because we are having some back signal interference here, it will be very nice if you can do this. Okay, so as I said to you in the beginning, we have a time to finish this session this afternoon, but it's very interesting questions here, presentation. So I would like to open for questions. So if one needs to ask something, maybe, yes, and yes, please.
I have one question from Carolina, because in fact I do work as well with microbiota.
Yeah.
And often when you have an inflammation or if you have dysbiosis, it's not so easy to have a good implantation of a bacteria you like to put back. But do you have some idea already or some data in your work with the patients if you have a good engraftment of a protein or back? Because often it's,
Yes, we still do not have, we are launching out our trial on FMT, so it's going to be open from November. But we have access to the data of the study that were published in Nature Medicine last year from Bertrand Roussy, and we are trying to explore how these signals to ameliorate the engraftment. But sure, we have the ideas that we have now is that the ideal is to have a knowledge of how is the gut microbiota composition of the recipient and the donor to have a better engraftment. But yes, we are trying to learn how to ameliorate.
Thanks.
Very nice talk for all of you. Continue on your talk about Akkermansia.
Akkermansia is good.
Yeah. So how is good? That's my question.
No, yeah.
What's the mechanism? Is just secreting something?
Nice question. We are working on the mechanism. We know that we have some metabolites that are associated with the good benefit, and what I currently know is that but now what we are trying to do is that in our prospective cohort we have more than the metagenomics data. So we have all the gut epithelial biomarkers, we have spectral cytometry from the patients, exactly to try to understand always the mechanisms. And sure, we have the clinical trials, so we are going to have the first results soon to try to understand how is it working, and if we really have the benefit to associate Akkermansia mucinifera, in this case is Marceliensis, a different type, of for patients that are Akkermansia negative as the baseline.
Thank you. We
Can I
have
ask a question?
Yes, please.
Even if I'm not an immunologist.
Oh, you're gonna learn.
I do my best.
I think you learned a lot this
Yeah.
today, so when you come
Thank you
back
for
to
the
France, it will be a
presentation. They
complete.
were amazing. Tiago, I have to ask you a question about pancreatic cancer.
Sure.
We've been seeing very interesting stuff in mouse model actually, because oncolytic virus infection, proper infection, has been associated in the past with those TLS. It was a good sign of efficacy. I was wondering maybe that might be one trigger for help maturing what you call the transition between immature to mature TLS. That might be associated with better response to immunotherapies, because just like me, that actually right now patients are not getting immunotherapies with pancreatic cancer. So what is your point of view about that? Have you heard about that?
No, I haven't heard about that. The only thing that we have the chance to investigate was the fact that when we microdissected these structures, so we need to have follicular dendritic cells over there to induce an immune response against the cancer cells. So we don't know the antigen origin of that. It might be something related to virus, but we are not quite sure about it. And we have no idea what tumor antigen should be involved with that. No, we don't have quite a lot of evidence behind that.
So might be worth playing with the mouse model we have in the lab.
With the mouse model?
Yeah.
No, we haven't had the chance to work with mouse models. No, we want to establish that in the laboratory, but we still didn't have the chance to do so. But
So
yeah,
we can
that
talk.
should be something. Yeah, the next stage, and it's getting more and more difficult to get samples from the patient. So yeah, it's a good idea to move to the mouse models for sure.
Thank you.
You're welcome.
I think Agnes has another question.
Okay, I also appreciate all the talks, of course. They were excellent. I have one question for Marco. You showed that when you do the treatment with inulin, you have more gamma delta T cells, if I understood well, and more L22. Do you know what cells are producing L22?
Yeah, so we have shown we didn't see a change in the number of gamma delta T cells, but we see that the phenotype is dependent of gamma delta T cells. So if we remove that, then we don't have the phenotype. The same thing with IL-22. But we have seen increased production of IL-22 by different populations. So we know that ILC3 cells are responding, Th17, and also gamma delta T cells. So now we are trying to connect this with the phenotype, which is the main point is increased proliferation in the crypts, and also to connect with the metabolite that we think is acting on the cells. So that's what we are doing now.
Okay, so I do have one question to Tati. So very interesting data, wonderful presentation from all of you. But when you showed the last data on melanoma, that you clearly show the ones that get free of the disease and the ones that, yes.
Yeah, gliomas.
Sorry, but I thought on the, yes. So the question is, this specifically, you see that this is a specific localization of a tumor, so very hard to get samples when the patients are alive. So how can we actually translate this in basic clinics that we can stratify these patients? I don't know if this mirrors what you find in blood samples or in specific type of cells, this whole methyloma
Yeah.
analysis that you...
No, you're right, specifically because you're talking about brain tumors, right? So it's the only way to get the samples are when they're surgically resected. So no, for this work, we are just finding and describing signatures and trying to understand what's really going on. But there's another group, part of the group that was trying to match what we found doing liquid biopsy to try to translate this into clinics, I guess. It would be more.
Okay.
Yeah, not specifically with that, but maybe there will be some cool results. So when I was in the US, I participate in this liquid biopsy consortium. So people, most of the people are in common with this group, so they're collecting prospectively samples from the blood and trying to find epigenomic markers circulating in the blood that can help with those questions.
Okay. So sorry that I said melanoma, because melanoma is the question for Carolina, but
No
I will ask
problem.
her. So, and I have another question for Tiago, because I am the chair, so I'm allowed to make questions.
You
many
do whatever you
questions.
want.
Yeah, so very nice that you found B cells, and part of my lab is on B cells. So do you think that these B cells are there as APC, or they're actually turning into plasma cells? So what's the whole of the B cells inside the TLS?
They are turning into plasma cells for sure.
Really?
Yeah, we have investigated all of this data, but we didn't have the chance to bring it here for the sake of
So
time.
you have the know the
didn't
quality of
give
the
me much time,
antibodies.
right? So yeah,
I
you
will.
just gave me 12 minutes, so I couldn't do more than that. But yeah, we have essentially everything there, Niels. The B cells are there, the germinal center B cells are there. They have the whole molecular programs of light and dark zones. They can in the dark zones, the B cells express AIT. They are inducing hypermutation, somatic hypermutation, class switch recombination. The plasma cells have been generated there, and they are secreting IgG against tumor cells.
Yeah, so it means that everything is coordinated by TFL cells. So the CD4, like follicular-like.
They're also
cells.
there, yeah. And the beauty of it is that we don't have to go to the draining lymph node anymore. Everything is happening in situ, so that's the beauty of it.
But you probably need a chronic inflammatory milieu.
We do need a chronic inflammatory process to induce the TLS formation and maturation for sure.
Okay, I just made this question. So you know by now that this is all about immunology.
Immunology.
Yeah, so my last question before we end, or I ask for questions to Carolina, is about the melanoma. So very nice when you showed that you have a fingerprint that you can predict patients that will have the recurrence or not. My question is, are you sure that those patients that you removed or you treated the melanoma, they are melanoma-free patients?
Yeah, I don't know. Actually, it was a not really old, but an old clinical trial, so you do not have information, for example, about circulating tumor cells or anything. They were stage 3, so locally advanced melanoma. Yeah, so I don't know. We have this validation cohort that we are trying to get, that they have the CTC, and also we are going to do metabolomics to try to confirm. But it was a negative trial, 2 arms, and we could see, it was interesting to see like this shift, like they started to relapse like 80 months after, and after 1 month, we already saw the metabolism that were shifting, like we can see before we can really see the disease.
Okay, we have to finish, so I have to thank you, all of you. It was a wonderful pitch presentation, so just applaud them. And then let's move on to our closing lecture. I have the pleasure to invite Professor Connie Markmanos. She is responsible for all the international and national collaboration at FAPESP. So she will give us an overview how FAPESP can actually support collaboration between both countries. And that's why you are here until now, because you have to listen to that and then to apply, because there is some ways to find new collaboration with our French colleague. So please, Connie.
Thank you very much. Já vai colocar. Didn't understand a word of it. Well, it's not true, but my area is animal breeding, not medicine. So I just want to give you a very quick overview of how FAPESP operates. And this is the really interesting part of this whole 2 days, because it's about the money. So, the rest doesn't really matter. This is just what talk about this. Just for those of you who are from abroad, the state of São Paulo is the most prosperous state in Brazil, responsible for about 42% of all the scientific production in Brazil. We're about the GDP of Sweden, the land area of the UK, and the population of Spain, so basically a country, although we're a country within a country. Like I said, responsible for 42% of all scientific publications in Brazil. If somebody is collaborating with somebody in Brazil, they're collaborating with São Paulo. Basically, you will see in almost all papers that if there is collaboration, it is with São Paulo State. We have only 3% of the geographic area, 20% of the population, but 32% of the GDP. So, and I think it's very important to mention that 1% of the GDP of the state of São Paulo comes directly to FAPESP. That makes us very different from all other financing agencies in the country, because once the money comes in, it doesn't go out again unless we give it for research. So therefore, it's not like, for example, the federal agencies, whereby they have to have a yearly budget that is decided by the government, and then each year they get a budget, but not necessarily get the money. We get the money. So that means sure that we can have long-term relationships and long-term programs. So as you're going to see, give you an idea of those in a little while. So here in São Paulo, we have 67 higher education institutions. They're federal, state, and private institutions, with 32 government institutions, including federal and state research institutes, healthcare institutions, and hospitals. We've already heard here today from some of the researchers that are in some of the hospitals, and the research that's being done in the hospitals in São Paulo State is excellent. 23 private non-profit research institutions and 10,000 companies. So there's a wide gamut of research being done, but most of it is done within the 67 higher education, mainly 7 institutions, which are OSP, UNESP, UNICAMP. The 3 federal, those are the 3 state universities, 3 federal universities, which are Unifesp, UFABC, and UFSCar, and ITA, which is the Institute for Aeronautics. The mission of São Paulo, of FAPESP, is to support research in all fields since 1962, 1% of tax revenues. We have around about between 16,000 and 20,000 new proposals every year. And we support grants in all areas, and I'll show you that in a minute. Our annual budget is around about 230 million euros. And most of that is in the life sciences, which is good news for you, right? But we have in other areas as well. Here we divide into 2 different areas, because not only do we finance science itself, but we also finance innovation. So we're a bit different from other agencies whereby these 2 are separated. We finance both together. As you may have noticed, those people are from São Paulo. Some of these the regulations have changed recently. So our regular projects are now 600,000 reais, which is around about 100,000 euros for 3 years, not including scholarships. So therefore, that is, if you think that the federal government, the universal projects are 50,000 with 10 researchers for 2 years, our basic projects are very different. Then we have the thematic projects, which are around about 3 million for 3 years, 3 to 5 years. Also, and then scholarships outside that. Because the question is too, if you don't have scholarships, then you don't have the workforce to do the work in the project. So you need both. And then one thing is very important. If all these requests have to come from researchers in São Paulo, but any basic project in São Paulo can have international collaboration in it. You don't have to wait for a call. You don't have to wait for something else. Is there going to be a call with INSERM? Is there going to be a call with France? No, you can ask for money already within the projects, but it has to be somebody that's in São Paulo state that asks for that, okay? So then we have other questions that I'm going to show you in a minute, and then we have innovation too that I'm going to show you some of the work and research. So we finance capacitation. Here we have the São Paulo School of Advanced Science, which lasts from 1 to 3 weeks. We have mentoring, and there's the site. We're also studying, doing research capacitation for young researchers, and then look at project preparation and manuscript preparation with Fulbright and with Elsevier and Springer, see how we can improve the quality of the written word in projects and in papers. So we have money for mobility, both incoming and outgoing. We support outgoing lecturers from a month to a year, postdocs, PhDs, masters, undergraduates. We have the sprint program that there are 3 calls a year. And we also, with France, we have a lectorado that we just signed recently. I think the call is open at the minute. And that's for teaching Portuguese in France. And then tomorrow we have a meeting with Itamaraty to discuss in other countries. Incoming, we have the São Paulo Excellence Chair, whereby the person has to be here for 12 weeks per year for 3 years. Visiting researchers, so a professor from another institution in France can come here from 2 weeks to a year. The Fulbright Chair, Young Researcher Awards, and TWAS, which is the Third World Academy of Sciences also. So therefore, there are lots of options here for mobility. And then inside our projects, we have, I divided them into 3 basic groups. The first is getting to know you, because obviously if you're going to have a long-term collaboration with somebody, the first thing you have to get to know them. So we can both do congresses, so people from São Paulo going to congresses, or somebody organizing an international congress here. For example, those of you that are here from France, we helped finance your coming here. So, and then also in organization of workshops, we also help in organization of workshops. So after you get to know somebody at a congress or a meeting or a workshop or whatever, and then you have to, let's go to the engagement phase, say, okay, let's get serious, So we have the sprint program, we go into mobility. We can also have some low seed funding for start-off projects inside our major projects, so that is another option. And then we get into the long-term commitment. So therefore, yes, this is somebody that I really want to have a relationship with for a long term. So I didn't put marriage. Originally, the slide had marriage on it, and decided to change it, I think, because it's more modern now to have a long-term commitment or whatever. So we have joint calls. So for example, we have INSERM, ANR, we have other options as well that we can joint calls together, and then we can, there are specific terms of those calls. The funding within the regular calls, such as our common projects, our thematic projects, our young researcher projects, etc. And then we have special programs, such as Biota, which is in biodiversity, QTIA, which is in artificial intelligence. We have a call open at the minute for artificial intelligence in medicine, oceans, etc. So there are lots of options. You just have to keep your eye on the site to know what's available at the time. So we've started also defining priorities. So we're looking towards major players worldwide, and obviously France is one of the major players worldwide. So France for FAPESP is a priority financer within for our future. And then we have other things like Global South, emerging regions, and multinational relationships that we already have. We also have these 10-year projects. So these are larger projects that are called Research and Innovation Development Centers. These last 10 years, and they have maybe 10 million or more reais looking towards them. About half of them are in health and disease processes, and within them you can ask for national or international financing. So loads of options here. There are also, and then you know, once a call goes out, then the researchers from São Paulo can ask for significant financing within these centers. And then, for example, one example is this center that works on CAR T cell therapy for cancer treatment. And it's the only public center in Brazil that actually offers CAR T therapy, and it was financed by FAPESP. We also have these centers for innovation, so whereby a business or pays half, and FAPESP pays the other half, and it's also 10 years. And this is an example for Embraer, which it's one of the largest regional airplane manufacturers in the world. It's now owned by Boeing, and together with ITA, which is the Institute for Aeronautics, they have this project looking towards solutions for autonomous flying, and SAFI, which is sustainable air fuel, etc. So here on the right is the list, and you see there are a lot of these centers that work with, for example, GlaxoSmithKline, or with in the area of biological control of plants and diseases, or artificial intelligence, etc. So there are options here too, whereby you can look for financing, and this is all on our site. And there's just an example of one that really worked. So as everybody knows, here in Brazil, we have alcohol at our petrol stations. So this is a hydrogen reformer at the petrol station. So you don't actually have to do the hydrogen, you don't have to transport hydrogen. You can put it in the car at the petrol station. So this is something that was developed from a FAPESP project recently. And so this is just a diagram of how that works. And now they're working on how you can do this reform in the car rather than at the petrol station. This small business research. We also have money for small businesses and startups. So maybe within your lab, you have some ideas for a startup, and here there's money that can do that. And here's the Institut Cell Therapy winner of Brazil Awards in 2023. Artificial intelligence, as I said, is becoming even more important. So FAPESP also looks after this very large call that was made late last year. And there are now 10 of these centers starting up. I think half of them are in São Paulo. We have these other special programs. Most of those are bottom-up. People have an idea and come to us with them. These are top-down. So there's the Amazon Plus 10, public policies, global climate change, renewable energy, biodiversity, quantum theories, South Atlantic, Antarctica, basic education, data science, and loads of them have interaction with the area of medical sciences. So last year, we had 52 calls, 28 international calls. This year already, we have much more than this. But this was just the end of the pandemic, so that we're still just getting started. But now, I think this year we'll probably have upwards of 50 international calls. So there are loads of opportunities. You just have to be aware of what's going on and make sure that you get your proposal in on time. Okay, so thank you very much. If anybody has any questions or... Leandro.
So thank you, everyone. It's just closing our workshop. I think we get together for these 2 days to know each other, to establish a new collaboration, partnership, to reinforce old strengths. I would like to thank everyone, the participants, the speakers, coordinators. We hope to do this next year in France this time, have the same people going, maybe Toulouse, maybe other cities. France is a very important, as Connie already pointed out, country for connection to Brazil. And I think we show it just for health, for subjects, how important it is for us. So we hope this continue. Thank you very much. And now it's time to work. Thank you very much, everyone.